Hello, dear students. This is Bushra Sayed, and today we are entering our semester 3. So, I welcome you all to semester 3 and the first online lecture of semester 3. Since this is the new semester, you should know certain points of syllabus. So, when we talk about zoology, there are total two papers that you need to study for zoology, 201 and 202. Each paper will be having total 4 units. And today we are going to study paper 201 unit number 4, the genetics part of it. In that we will be covering three topics: paleotropism, duplicate genes and multiple genes. So let's begin. Paleotropism. You all have already studied this in, I guess, 12th standard. So we all know that Mendelian genetics says that a specific gene produces one phenotypic character, but we know that there are certain non-Mendelian phenomena which didn't follow the Mendelian genetics, one of which is this paleotropism. So there are certain genes which produce several side effects besides their own phenotypic trait, like they produce a major phenotypic trait. Besides that, they have some other effects on the body, that we say that they are giving more than one phenotypic trait. They are influencing more than one phenotypic trait. Here in the diagram, you will be able to see that, yeah. So you can see in the diagram that there are total three genes, and each gene is influencing more than one phenotype, right? So here it is given that one single gene is influencing two phenotypes, but it could be more than two. It could be two, it could be three, it could be many. Moving on towards the definition of paleotropism. It is the phenomenon in which a single major gene influencing more than one character is known as paleotropism, and those genes which does the paleotropism effect are known as paleotropic genes. Mechanism of paleotropy in most cases is the effect of a gene on metabolic pathways that contribute to different phenotypes. What does this say? That if a particular gene resulting into a protein, we know that genes results into certain proteins, so these proteins have some other phenotypic effect. So this particular gene will be influencing only one protein, but this single protein will be having different phenotypic effects. See in the diagram, if you can see that this is a single gene. Due to paleotropy, what happens is it gives rise to different traits, different phenotypic traits. We'll study one by one through examples. So our first example is phenylketonuria, PKU. What it is? It is a mutation in a single gene that codes for enzyme phenylalanine hydroxylase. So mutation, that means a change, a harmful change. So the harmful change that is taking place in the gene that codes for phenylalanine hydroxylase. This enzyme particularly converts phenylalanine to tyrosine. So if this enzyme is mutated, then phenylalanine will not be able to convert into tyrosine. So because of that, what will happen? Phenylalanine will accumulate in the body and it will show the side effects. What side effects? Apart from phenylketonuria, the major effect, it will be having mental retardation. It will be having other symptoms such as widely spaced incisors, pigmented patches on skin. So these are the different phenotypic traits. Not only one trait will be shown, but there are many different traits are being seen here. Second example, if we see, is the recessive gene for vestigial wings in Drosophila. So the gene, which is the recessive gene, we know the recessive genes are the small letter ones that we usually indicate. So the recessive genes for vestigial wings usually have, the Drosophila have vestigial wings, right? Vestigial wings are basically those who are not normally functioning wings of Drosophila, right? So that is the actual phenotypic trait of that particular, these particular recessive genes, that the Drosophila will be having vestigial wings. But apart from that, that particular Drosophila with vestigial wings will be having impaired reproductive organs, will be having reduced egg production and bristles on the wings. So apart from the vestigial wing, formation of the vestigial wings, they will be having other phenotypic traits. Okay, that is the second example for pleiotropy. Coming to the third example, that is the sickle cell disease. We call it as sickle cell anemia. We know that beta globin gene mutation leads to sickle cell anemia. That is the formation of a sickle RBC. Okay, the shape of RBC changes. We know that. So apart from change in the structure of RBC, this will be we know that it will be, the major effect will be anemia. Okay, apart from anemia, what it will lead to? It will lead to blindness, it will lead to liver failure, it will lead to heart diseases, which could lead to heart attack, right? So this is basically pleiotropy. What does it say? Is that a single gene will be influencing more than one phenotypic trait, and these are the examples that you need to study. Next one, that is the duplicate genes. So here in the diagram you see that gene 1, there are total 2 genes. Okay, so gene 1 and gene 2 both will be influencing one particular phenotypic trait. It is one kind of opposite of pleiotropy that we have studied, that gene, there it was one particular gene and several phenotypic traits. Here there will be one phenotypic trait which are being influenced by two different genes. That is why we call them as duplicate genes. Now, what is its definition? When two pairs of genes determine the same or nearly the same phenotype, so that either of the two genes is able to produce the character. See, either of the two genes is able to produce the character. It means that if only gene 1 is present, the dominant gene 1, I'm talking about the dominant gene 1. If the dominant gene 1 is only present, then also this phenotypic trait will be expressed. It is independent of whether these two genes are present or not. Even if one single gene, one single dominant gene out of the two, if they are present, it will be showing this particular phenotype, the same phenotype. Okay, which means that duplicate genes do not have cumulative effect. Cumulative effect means the additive effect. Okay, when two different genes join, combine and give one particular phenotypic trait, it is not like that. It is that separately also they can give the same phenotype, together also and separately also. Okay, if both the genes are dominant genes, right, if both the dominant genes are there, then it will give the same phenotype. Even if one dominant gene is present, then also it will be giving the same phenotypic trait. Okay, so we'll study with the help of one example which is given in your syllabus. Seed capsule shape in shepherd's purse. Okay, shepherd's purse is nothing but it is a plant. Okay, the seed capsules, when we talk about the seed capsule, that is the covering of this seed. We can also call it as a fruit, right? So, it is of two different shapes. It could be triangular, it could be elongated. We know that we are studying genetics, so we will be crossing between the different characters, right? So if we cross between the triangular and the elongated seeds or the plants, the plants having the triangular as well as elongated uh, seed capsule. So what will happen? The F2 generation, from this F1 generation, the F2 generation will be giving triangular and elongated shape in the ratio of 15:1. It means that there will be 15 one particular character and one will be of other particular character. Okay, definitely we will be studying with the help of the dihybrid cross. Before that we need to specify the genes. Okay, So suppose T and V, the dominant T and dominant V controls the triangular character. Look, and they both are located on different chromosomes. Okay, they are located on different loci of two different chromosomes. Since they are duplicate genes are located on different location of different chromosomes. Okay, they are not linked with each other in any manner. Still they give rise to the same phenotypic trait. Okay, so yes, so suppose we have the dominant gene T and V, which controls the triangular character located on two different loci of two different chromosomes. Singly or collectively, it means if there is only one dominant gene, or both of them will give rise to the same phenotype. What if the dominant genes are absent? Then there will be the presence of recessive genes. That recessive gene will give rise to elongated or ovate seed pods or seed capsule. Okay. Which are the recessive genes? We know that small t and small v will be the recessive genes which will give rise to elongated or ovate pods or fruit seed capsules. Yeah, so here is our digahybrid cross, triangular and the ovate. We know that ovate or elongated will not have any dominant gene. Presence of a single dominant gene will be a triangular shape seed capsule. So F1 generation will be having all triangular. Here we know that at the end one is the only one in which we don't get any dominant allele or gene. So that is why there will be only one elongated or ovate seed, whereas the other 15 will be triangular. That is why the phenotypic ratio that we get in duplicate genes is 15 is to 1. Okay, which is not same as the Mendelian genetics. We know that in Mendelian genetics we used to get the phenotypic ratio as 9 is to 3 is to 3 is to 1. But since this is a concept of non-Mendelian genetics, here it gives rise to the phenotypic ratio of 15 is to 1. Okay, so coming to the third one, that is the multiple genes. Okay, when we talk about multiple genes, see, we have already studied about multiple genes in our semester 2, right? The multiple genes, we have studied about the multiple alleles, the blood groups, right? We have seen the multiple genes. What does it say? Is that there are different other genes which are responsible for a particular trait. Okay, their additional effect. If all the genes are present, then only one particular phenotypic trait will be there. Otherwise, it will be a defective one. So here in the diagram, you can easily make out, I have taken 4 genes. See, multiple genes could be more than 4 also. So there are total 4 genes that I have taken. Cumulative effect, the additive effect of all these genes will be resulting into one similar phenotypic trait. We have talked about the blood groups, right? The blood groups, there were several genes which are responsible for a particular blood group. Okay, so like this only, this is the same phenomenon, but here we will be studying some other example, because in our syllabus some other example is given, which is the tryptophan synthesis pathway. Tryptophan is nothing but it is an amino acid. So tryptophan synthesis pathway in E. coli is the example that we need to study for multiple genes. Yeah. So first you need to know what is tryptophan synthetic pathway. It means that how tryptophan is formed in the bacterium E. coli, right? So how it is formed? Choriamic acid, it is the precursor in the presence of gene 1. We know that gene is responsible for the expression of some protein. Which protein? This protein, enzyme 1. Okay. So gene 1 will be expressing enzyme 1. In the presence of enzyme 1, it will give rise to anthranylic acid. Okay, we know that enzyme and substrate results into a product. Okay, here is your enzyme, here is your substrate, which give rise to the anthranylic acid, that is the product. Okay, Coming to the next step, okay, we have to form tryptophan. Okay, so the next step involves the other gene. This is gene 1, this is gene 2. It involves the other gene which will express enzyme 2. In the presence of enzyme 2 and substrate anthranilic acid, there will be the formation of CDRP, that is carboxyphenylamino deoxyribulase phosphate. Okay, coming to the next step, gene 3. Okay, gene 1, 2 and 3, we are on the third gene. Gene 3 will give rise to enzyme 3. In the presence of enzyme 3, this CDRP will be converted into indole glycerol phosphate. Okay, so tryptophan is not formed yet. Now in the presence of gene 4, which will be giving rise to enzyme 4 or expressing enzyme 4, in the presence of enzyme 4 and indole glycerol phosphate, there will be the production of tryptophan. So this is the whole tryptophan synthetic pathway in E. coli bacterium. So multiple genes. Here are 1, 2, 3, 4. Total 4 genes are there which are responsible for the formation of a single amino acid, that is tryptophan. If any one gene, for example, if there is any one gene which is absent or defective, or any one enzyme which is not completely formed or defective, or it is absent, then it will lead to the failure of the formation of tryptophan. Okay, so this is about the multiple genes. We know that multiple genes, we have studied various examples in the previous semester. The first one was multiple alleles, the ABO blood group. Then the polygenic inheritance, if you know, that is a color of humans, okay, the skin color of humans. This is the third example of multiple genes. So it is very easy to study and very easy to understand. Yeah, so we have studied total 3 concepts today. Hope you understood it. Okay, if there is any query, you can contact me on my mail ID, or you can also WhatsApp me. Thank you and good luck.